In Lecture 8, we will be looking at heredity. We will look at the process of creating sperm and egg. It's part of what we call meiosis, and we'll compare that with what we've learned before in mitosis. We will also be solving various genetic problems. For example, if you have brown eyes and your spouse have blue eyes, what's the probability that your child is going to have blue eyes? And we will be looking at the basis of inheritance, the concept of dominance and recessive. So a lot of stuff to go through. Let's get started. Let's start with meiosis. So the purpose of meiosis is to allow sexual reproduction. In lecture 6, I think, we learned about asexual reproduction, where you only need one individual and all the offsprings are going to be genetically identical to the original parent. But in sexual reproduction, it involves the fusion of reproductive cells from two parents. So in humans, the reproductive cells are basically sperm and eggs. And the sperm will fertilize the egg and it will create the baby. In the process of meiosis, we are going to be cutting the amount of chromosomes by half, and so the sperm and egg only carries 23 chromosomes as opposed to 46 that are found in the regular bodily cells. In order for us to be able to do that, meiosis is the basis of sexual reproduction. So human cells are considered as diploid cells. In diploid cells, we have two sets of each chromosomes. So we know that already, right? We know that there are 33 pairs of chromosomes, and half of it is going to be inherited from each parent, meaning there are going to be 2 chromosome 1, there are going to be 2 chromosome 2, and so on and so forth, all the way to chromosome 22. And then for chromosome 23 is either XX for female or XY for male. And these are going to be homologous, we call them, because they contain the same set of genetic information, but perhaps different versions along the way. So for each chromosome, you have 2 copies, and therefore collectively we have 2 sets of the entire chromosome 1 through 23, and that's what makes us diploid. Gametes, on the other hand, human gametes are the sex cells, the sperm and eggs. They are haploid, and they only have 1 set of the chromosome. And so when fertilization occurs, when the sperm and egg joins together, it creates what's called a zygote. The zygote starts off as a single cell, and the sperm contains 23 chromosomes, the egg contains 23 chromosomes, but when they fuse together to form the zygote will now have 46 chromosomes. Okay, so sperm and egg, they are haploid, and the zygote will be diploid. Okay. And that's how we got our genetic material, half from the sperm of the father and half from the egg of the mother. This zygote starts off as a single cell, but then it will rapidly divide using mitosis following fertilization, and after about 2 weeks or so, the zygote will eventually becomes the embryo. And then the embryo is going to grow into the fetus, which, as we know, will become the baby. The process of sexual reproduction begins with meiosis, the process of creating the sperm cells and the egg cells. And in order to do that, we start off with what's called germ cells. Okay, so germ cells, the name is a little bit misleading for some people. They hear the word germ, they think of like bacteria and dirty stuff. But in the context of what we are discussing, germ cells refers to the cells that are found in the gonads. Okay, so gonads are the reproductive organs in humans. For the male, the reproductive gonads would be the testicles, the testes here, and for female, it would be the ovaries. So within the gonads, within the testes and the ovaries, there will be these diploid germ cells. Okay, and then the germ cells is going to undergo meiosis, and in the process, it will create haploid gametes. Germ cells are also found in other organisms like plants, except for plants, they would have both reproductive structures in the same same organism. So they are a little bit different than us. They are called hermaphrodites, meaning they have both male and female reproductive organs. So in the plants, the antler is the male reproductive organs. That's where the sperm is going to be produced, but we don't really call them sperm, right? We call them pollens. And over here, that's where the ovary is going to be. So plants can self-pollinize, self-fertilize rather, or it can rely on pollination to cross-fertilize. Here is an overview of meiosis. We start with the parent cell here, and the parent cell is diploid because it contains two sets of chromosomes. So we have one chromosome from mom and one chromosome from dad, the maternal chromosome and the paternal chromosomes. For simplicity, we are only showing two chromosomes here, but if we are talking about normal human cells, there would be 46 chromosomes in total, 23 from each parent. So over here, this would be your germ cells that is going to be undergoing meiosis to become a gamete. Okay, and the gamete is either going to be the sperm cell in male or the egg cell in female. So here we, again, for simplicity, are just showing two chromosomes. So two chromosomes. And what happens is, before we have the first division, before we have the meiosis I, we are going to duplicate our chromosomes. We are going to undergo DNA replication to make a copy of each of these chromosomes. So afterwards, you would have an exact copy of the original, and they are going to be held together at the centromere. Now here is where things could potentially get a little bit confusing. If I ask you, how many chromosomes do you see here? You might be tempted to say there are 4 chromosomes, but the correct answer is actually still 2 chromosomes. Okay, so biologists have agreed that if you have 2 chromosomes that have been duplicated and are held together at the centromere, this whole thing is still considered as 1 chromosome. So this whole chromosome would consist of 2 individual sister chromatids, and then there is going to be the second chromosome, and this one would also have 2 sister chromatids. Okay, so we started with 2 chromosomes, and after DNA replication, after the chromosome duplication rather, you will still have 2 chromosomes, but now you will have 4 sister chromatids. Okay, so 1, 2, 3, 4 here, right? And we started with a diploid cell, so that's 2n, right? 2 sets of chromosome, and here we still have 2 sets of chromosomes. Okay, the red and the red are considered as the exact same set, so for that reason, we will still say there is only 2 different sets here: the red set from mom and the blue set from dad. Okay, then what's going to happen is meiosis I is going to separate these chromosomes. So after the first division, we now have 1 chromosome. 1 chromosome, right? So we essentially reduce the number of chromosomes by half. And at this point, if you count the chromatids, there will be 2 sister chromatids. And we are down to 1 set of chromosome, right? Over here we have the red set and the blue set, so that's diploid. But now we are down to 1 set, so this is going to be 1n, and we call this haploid. Okay, and then meiosis II is going to have a second division, and now we are going to, in the final products, have 1 chromosome. 1 chromosome, and this is going to be 1 chromatids, right? And we essentially don't have to write that anymore. And each individual product, gametes, will have 1n. Right? So this is also haploid. So the process of meiosis, as we just saw, consists of 2 consecutive divisions, and they're just called meiosis I and meiosis II, and the daughter cells are going to be genetically different than the parent cell and only contain half of the genetic materials. in order for us to create variations in the gametes, genetic variation that is, we have to do something called crossing over. So let me just recap this for you a little bit. If you think about your cells, okay, your germ cells, okay, this is the one that's going to undergo cell division. What you will have, as we keep on mentioning, is that there is going to be one maternal chromosome. This is from your mother, and then you're going to have one paternal chromosomes, okay, and then this is going to happen for the entire 23 pairs. But to keep it simple, we're just going to do one pair to look at it, and so this is from your mother, and this is from your father, and we call them homologous chromosomes. Homologous chromosomes. And homologous chromosomes are chromosomes that are going to be same size, same length, and they are going to contain the genetic information in the same order. However, the version of the gene might be different. Okay. Perhaps I'll put an other pair here just so you have an idea. So this might be like chromosome 1 and chromosome 1, and then this would be smaller, right? As you go higher in the chromosome number, the size of the chromosome actually gets smaller. So this might be chromosome, I don't know, 20 or something like that. Okay, but in reality, you would have a total of 23 pairs. Okay, so this is chromosome 1, chromosome 1. They are homologous. Chromosome 22 from mom, chromosome 22 from dad. They are also homologous. And then what happens is you will do DNA replication. Right, so the stuff that we learn in lecture 6, right, with the whole semi-conservative model and the helicase and all that stuff. Okay, and after DNA replication, what you will have is you will have 2 of each homologous chromosome now, right? So let me try to get this color. So there's going to be one original chromosome 1 from mom, but after DNA replication, you're going to have a second one, okay? And then you will have one chromosome 22 and a copy of it, and then for the paternal copy, that would be the same case, okay? So let me just put some numbers here for you. Again, this was chromosome 1 from mom, chromosome 1 from dad, from like your mom and your dad, right? This is your cell, and then this is chromosome 22. I said, right? Chromosome 22, and you would have, you know, the rest of them, which I'm not showing here. So after DNA replication, they will be held together at the centromere, right? So this part where the 2 copies are stuck together, that's the centromere. That's where the spindle fibers will attach to when you do cell division, right? So now there is a chromosome 1 and the exact same copy, and here, 22, 22, 22, 22, like that. Okay, so this and this, that's called the sister chromatids, right? Okay, so what happens is in mitosis, the homologous sister chromatids are not going to find each other. Okay, like this X is going to be independent of the paternal one. Okay, they're not going to be in the same place. But in meiosis, they are actually going to come together and form what we call a tetrad. Okay, tetrad. Okay, tetrad. And when they form the tetrad, what happens is they can do something called crossing over. In the process of crossing over, a little bit of the red chromosome will exchange with a little bit of the blue chromosome. Okay, and this exchange will happen along the entire way, and it could happen between any of the 2 chromosomes. Okay, so it could happen between this chromosome and this chromosome, which is what's shown here. It could happen between this chromosome and that chromosome. Okay, but for simplicity, they are only showing one small crossing over here. Okay, so that involves the exchange of genetic information between the homologous chromosomes, and you end up having like a mix of genetic material. So it will no longer be exclusively maternal genetic material or exclusively paternal genetic material. Right now, it's a little bit of red, a little bit of blue, and then over here a little bit blue and a little bit of red. Okay, and again, in reality, it's not just going to be one small section. It's more like, you know, blue and red all mixed together in the end. And this process is important because it allows for genetic variation to arise. We call this genetic recombination. Let's start with meiosis I. In meiosis I, we have the prophase I, metaphase I, anaphase I, and telophase I. It's pretty much the same thing that's going on in mitosis with a few key differences. So let's start with prophase I. In prophase I, you would have the chromosome starting to form, right? They used to be chromatins, but now because we're ready to divide, they will condense further to form the chromosomes. DNA replication would have been completed at this point, and as I just mentioned in the previous slide, the tetrads are going to form. Okay? And a tetrad is basically the homologous chromosomes coming together, right? This does not happen in mitosis. In mitosis, the big blue one here and the big red one here is going to be completely separate. They're not going to be associated with each other. However, in meiosis I, in prophase I, they will find each other. Okay, chromosome 2 will find chromosome 2, chromosome 5 will find chromosome 5, and they will come together to form this tetrad. And by forming the tetrad, it allows them to perform the crossing over that we discussed on the previous slide. Also, there is going to be these centrioles. They're going to be moving to the opposite side of the cell, and as they do that, they will be producing the spindle fibers. And of course, the nuclear envelope is going to dissolve in prophase I, and that allows the chromosome to come out from the nucleus. In metaphase I, the tetrads will line up in the middle of the cell, and we will have the spindle fibers attaching to the centromeres of each of the tetrads pair, and they will do a tug of war that allows them to all line up in the middle of the cell. Next, we have anaphase, where the homologous chromosomes of each tetrad are pulled apart by spindle fibers, and they will be pulled to the opposite side of the cell. And afterwards, there will be telophase I, where the two separated chromosomes are going to be surrounded by a nuclear envelope, and then that concludes the end of telophase I, meiosis I, in fact. Afterwards, we have prophase II, and in meiosis II, the steps are almost exactly the same as what happens in mitosis. So in prophase II, you will dissolve the nuclear envelope again. The spindle fibers will form one more time with the centromeres moving to the opposite side. Chromosomes will condense once again, but the key difference here is there will be no DNA replication. Okay? It just continues from where you left off in meiosis I. The chromosomes are going to be lining up in the middle with the spindle fibers pulling them at the centromere, and then in anaphase II, the sister chromatids will now be separated by the spindle fibers, and they will move towards the opposite side of the cell. And finally, you are going to be able to form a total of 4 cells, and the nuclear envelope will surround the genetic material, and you end up having half of the genetic material that you started with, and also they are going to be genetically different because of the crossing over step that occurred in prophase I. So I will post a link to animation of this. It will be much easier to see it, in terms of a process rather than going through the slides like this. Here's a table summarizing the difference between mitosis and meiosis. Let's just go through it together. In mitosis, they produce genetically identical daughter cells, and there are going to be 2 daughter cells per mitotic division. In meiosis, you are going to be producing genetically different daughter cells, and typically speaking, we produce up to 4 cells per meiotic division. Okay, I say up to 4 because there's a little bit of difference between what happens in male and female, and we're going to get to that in a second. Mitosis produce diploid daughter cells. Each of the daughter cells will have 2 sets of the chromosomes, whereas in meiosis, you are producing haploid daughter cells, which means each of the cell, the sperm and the egg, will only contain half of the genetic material, 1 set, right? So 23 as opposed to 46 over here. And in in mitosis, it is a process that will take place in most somatic cells. Somatic cells are your bodily cells, basically everything except for the sperm and egg. Not all cells is going to be able to divide. We talk about how brain cells don't divide, but if your cells are going to divide at all, they will be doing mitosis. Whereas the meiosis will only take place in special germ cells that are found in the gonads, the testis in male, and the ovaries in female. In mitosis, particularly in metaphase, the sister chromatids line up in the cell center. However, in meiosis, in metaphase 1, it is the tetrad that lines up in the middle. I'll show you a graphical diagram on the next slide to show you exactly how this would look differently in mitosis and meiosis. And also in anaphase, the sister chromatids will separate, but in meiosis, the homologous chromosome separates during anaphase 1. So that's what it will look like in a diagram. We have mitosis on the left here, and you can see in prophase, the homologous chromosomes, they do not find each other. Okay, they do not find each other. They're just gonna be floating around randomly. But in meiosis, the homologous chromosomes will find each other to form the tetrad. Okay, forming the tetrad allows you to do crossing over, and that results in genetic recombination. That causes your genes to be shuffled. Okay, no gene shuffling in mitosis. In metaphase, you can see the individual sister chromatids pair will line up in the middle, but in metaphase 1 of meiosis, the tetrad lines up. Okay, so 4 chromosomes lined up in the middle. Here, 2 at a time. And then of course, there is only one division in mitosis, but there are 2 divisions in meiosis. There's no picture here showing it, but following the metaphase, you can see in anaphase and telophase, the sister chromatids will be pulled apart. Okay, whereas over here, the entire pair will be pulled apart. So the homologous chromosomes will be pulled apart from the tetrad. Here, the sister chromatids will be pulled apart by the from its copy. Right, and in the end, the mitosis produce 2 daughter cells. Each will be diploid, whereas meiosis, up to 4 daughter cells, each will be haploid. So there you have it, the comparison between mitosis and meiosis. Meiosis in humans are actually referred to more correctly as gametogenesis. Gametogenesis is the production of gametes. Gameto referring to the gametes, sperm and egg. Genesis is to create. So we are creating gametes in the gonads, which would be the testis in male and ovaries in female. So in the testis, we more specifically call it spermatogenesis because you're producing sperm, and in females, we are going to be doing oogenesis because an egg is called an oocyte. Right? So that's why it's called oogenesis, and it's going to be a little bit different than the normal meiosis that you will see. Let's start with spermatogenesis. Over here on the left, we start with what we call a spermatocyte. That would be the germ cell. The germ cell is the diploid cell that will be undergoing meiosis, or in this case, spermatogenesis. After the first meiotic division, you would have 2 cells, and then you would have a second meiotic division, resulting in 4 sperms with 23 chromosomes in each. So that's pretty much the normal meiosis that we've seen. In female is a little bit different for 2 reasons. At first, we start with the oocyte, okay, and what's going to happen is that is going to divide just like normally. However, the meiotic division in telophase is not going to result in even division of the cytoplasm. Okay, one of the daughter cell will actually get much more cytoplasm than the other one, and so the one that didn't get as much cytoplasm is something we call polar body. Okay, so the polar body will not fully mature into an egg. Okay, it just kind of divide one more time and then it's gonna be gone. Right? And in the second meiosis, the same thing happened. You have uneven cytoplasm division. In telophase 2, so in each oogenesis, there's going to be just one single egg being produced. The rest are what we call polar bodies, and they are not going to be fully mature into an egg. So that's the first difference between spermatogenesis and oogenesis, and that's why the previous slide says, oogenesis is quote unquote complex meiosis. It's also complex in another way, and that's because of the timing. So in spermatogenesis, it basically takes place continuously after the male reach puberty, and the testis can produce up to 200 to 300 million sperms per day, but only about half of those will become viable. Okay, the rest are going to be reabsorbed and not be able to be fully functional. So that happens continuously, right? But for oogenesis, it actually starts happening while the female is still in embryonic development. Okay, so before birth, the oocyte will do meiosis, but then it will stop at prophase 1. Okay, and the egg will be suspended in prophase 1 until the female reaches puberty. After puberty, once a month, right, the one of those suspended eggs or cell at this point is going to progress from prophase 1 all the way to metaphase 2 during ovulation. Okay, and if fertilization does not happen, then that's it. The egg's gonna disintegrate and pass out as part of the menses. However, if fertilization takes place, then meiosis 2 will be completed after fertilization. So you can see the timeline is very different in oogenesis and spermatogenesis. Spermatogenesis happens on a daily basis, and it's going to be completed on a daily basis after puberty, that is. But oogenesis kind of span almost as early as the embryonic stage all the way to until the woman conceive a child, and it will be completed after fertilization. Okay, so big difference there between spermatogenesis and oogenesis. There is a potential problem that could occur with meiosis, and that's something called nondisjunction. Okay? In nondisjunction, the chromosome pair fails to separate, and this could happen either in anaphase 1, right, or anaphase 2. Okay? If it happens in anaphase 1 during meiosis 1, then 2 of the final gametes will have additional chromosome, while 2 of the final gametes will be missing a chromosome. Okay? Whereas if you have a nondisjunction that happens in meiosis 2, 1 of the final gametes will have additional chromosomes, and 1 will be missing a chromosome. So in a sense, a nondisjunction in meiosis 2 is less damaging than meiosis 1, okay? Because in here, all 4 of the final gametes are going to contain error. Either you have extra chromosomes or you have missing chromosomes. But if it happens in meiosis 2, at least 50% of the gametes are going to be okay. So why is it different, and how does it actually look? Well, here is a diagram that shows you what happens if the nondisjunction happens at different parts of meiosis. Here you can see the homologous chromosomes forming a tetrad at the end of prophase 1, and those homologous chromosomes are going to separate in anaphase 1, and that's what they will look like if there are no errors. And here in anaphase 2, the individual sister chromatids are supposed to separate from each other. So on the right here, the normal separation occurs, so each of the daughter cells, the gametes, will have 23 chromosomes, which is what's expected. However, if nondisjunction occurs, meaning these chromosomes do not separate, then the entire set will go to one cell and not the other. So you will end up having a cell with an extra chromosome, and then the other one will be missing one chromosome. Okay. If nondisjunction occurs earlier on in meiosis 1, then the problem is a little bit more severe, because instead of having the homologous chromosomes separating from each other, the entire tetrad will move to one cell. Okay, and then afterwards, if you do one more normal division, then you end up having 2 gametes with 24 chromosomes, and then here the one that's missing the tetrad will end up having gametes that will have 1 less chromosome. And that could be a problem because if you fertilize an egg cell that has an extra chromosome, for example, then the zygote is going to have 2n plus 1, right? It's going to have 47 chromosome as opposed to 46, because the sperm would be carrying 23 chromosomes, and then the eggs n plus 1 means it would be carrying 24 chromosome, and the zygote will now have 23 plus 24, resulting in 47 chromosome, and depending on which chromosome is extra, we will say is trisomy something. Okay, so there is trisomy 18, meaning there is an extra chromosome 18, right? And trisomy 21 would mean that there is an extra chromosome 21. The fertilization of gametes with a missing chromosome are usually lethal, so they are not going to form a zygote at all. So these cells, right, this one over here, this one, even if they get fertilized, they're not going to make it. Okay, so that's good in a sense because there will not be any chromosome abnormalities in the baby. But if you have extra chromosomes, then that's where problems are going to arise. So to determine whether or not someone has abnormal chromosomes, we can do something called karyotype. Karyotype is an orderly arrangement of chromosomes, and we can use it to screen for certain genetic orders when the fetus is growing. Here you can see we arrange the chromosomes by size. The longest one or the biggest one is chromosome 1, and the size goes down as we go up the numbers, with the exception of the sex chromosome. So remember, chromosome 1 to 22, we call them autosomal chromosomes. They encodes for everything about us except for the gender, but X and Y that would encodes for the gender. So XY here is a male in this particular karyotype. So we can use it to screen for things like trisomy 21, which is Down syndrome. There is an extra chromosome 21. There is something called Turner syndrome, which only has one X chromosome, right? So we call that monosomy X. So missing a chromosome in one of the autosomes are usually lethal, but if you are just missing an X, if you just have only one X chromosome and you don't have the other sex chromosome, that seems to be relatively okay in terms of development, but the person will end up having Turner syndrome. So in order for us to do karyotypes, we have to gather a little bit of the cell from the baby, from the growing fetus, and there are two ways you can do this. The first one is what we call amniocentesis. In amniocentesis, there is going to be a needle that is going to be injected, close to where the belly button is of the mother, and this whole process is going to be guided by ultrasound. So the person who's performing the procedure will know where the needle goes. Right? You don't want to be like injecting. Hitting the baby or anything with the needle, right? And the fluid that that surrounds the baby in the uterus is called the amniotic fluid. When the woman gives birth, you hear people say they would break the water, right? So that water that comes out, that's actually the amniotic fluid. So the amniotic fluid contains cells from the baby. So if you take a sample of the amniotic fluid, then you can harness the baby cell and you can perform a karyotype at about 15 to 20th week of pregnancy, and based on the analysis that would give you information about the baby. So in this case, it shows that there's an extra chromosome over here. Another option you can do is something called CVS, chorionic villus sampling. This is alternative to amniocentesis for prenatal diagnosis. And instead of taking a sample of the amniotic fluid, we are going to be taking a small sample of the tissue called the chorionic villus. which is part of the placenta. You can think of it as the baby side of the placenta, and it's derived from the same tissue cells as the baby. So it's going to have the same genetic material, and that involves inserting a catheter through the vagina, and the whole process will of course be guided by ultrasound, and you can perform this as early as 9 weeks and all the way up to 14 weeks of pregnancy. One big advantage of CVS is that you can do it much earlier in pregnancy than amniocentesis, and so if sometimes there are some detrimental genetic disorder that has been identified through the karyotype, then the mother can make a choice, perhaps involving abortion. So both amniocentesis and CVS are extremely common procedures, but there is of course a chance of miscarriage associated with them, because after all you are kind of disturbing the fetus, but generally speaking, they are a relatively safe procedure that offers genetic screening for the fetus. So there are some genetic disorders related to the sex chromosomes. Either you have extra copies of them, or you're missing a copy of them. And the first one is called the Klinefelter syndrome. In Klinefelter syndrome, the father of the individual would have to have a nondisjunction that caused the X and the Y chromosome of the father to not separate during meiosis. And if that sperm carrying both X and Y fertilizes an egg from the mother, then that creates the XXY. And this can also occur if the non-disjunction happens in the mother of the person, where there is no separation for the X chromosome. So the egg would be XX, and now it gets fertilized by a sperm carrying the Y chromosome. In either case, the primary feature of someone with Klinefelter is sterility. That means they're not going to be able to have babies of their own. Often, other symptoms are kind of subtle though, okay, and people might not even realize they are affected. And children with Klinefelter syndrome might have some learning disabilities or delayed speech and development related to language. But for the most part, many people have mild symptoms. Another one is called Turner syndrome. In Turner syndrome, we call it the XO genotype. Genotype is basically the genetic makeup of the individual, and that's as a result of missing an X chromosome in female, and that could cause a variety of medical and developmental problems, such as short height, even in adulthood, failure of ovaries to develop, and so these individuals might also be sterile, and there could be heart defects as well. So you can see in the karyotype here, there's only one X chromosome. YO, which is just one Y chromosome with nothing else, that is non-viable. Okay, it will not be able to develop at all. So we don't see people with that genotype. The next one is Jacob's syndrome. In Jacob's syndrome, we have XYY. So in this case, the non-disjunction has to have occurred in the father of the individual, where the Y chromosomes are not separated from each other during meiosis, and then that sperm carrying two Y chromosomes fertilize an egg carrying an X. Symptoms may include being taller than average, acne, and an increased risk of learning problems. But again, otherwise, the person is, for the most part, quite normal, and they can actually have children as well. So fertility is not affected in this case. So that's it for this video. We have covered all the that we have to for meiosis. In the next video, we will be looking at the molecular basis of heredity. Okay, and there's going to be a lot of problem solving involved. So come back when you are refreshed, and we will continue with the lesson. Thanks for watching.